Hello. So we are studying the microbiology book by Prescott, the scientist, and we are currently at the chapter 1, the history and scope of microbiology. So we were at the landmarks in the history, and we are currently in the after the 1945, or sorry, not the atomic bomb, but the 1944 Waxman discovered streptomycin. Okay, we are here at 1944 that the Waxman discovered the streptomycin. Streptomycin is the okay. I was just checking if the live was working. It's a problem for me if it doesn't work. So anyway, wait. Let me just change the setting in case if something gets wrong, we should know. So where is it? Where is it? Let's make it low, and let me just bring out the chat. So in case wait, so in case if anyone so the thing is, I don't mind. Where is it? Live. So I'm just bringing out, where is it? My live. Where's my live? Here. I just want to bring out the chat. Yeah, I'm gonna pin this. Wait, it's taking a bit time. Yeah, here. Let's stop this. The pop out chat. Yeah, sorry for the delay. I just want to see if anyone, just in case, text me. So yes, we are here. Okay, let's go back here. Let's start studying without any other, without any delay. I hope there is no, information. Wait. Let me just check out in case if something goes wrong. So anyway, we are here at the 1944. Did I turn that off? Yes. The Waxman, the scientist, discovered the streptomycin, right? Streptomycin is an antibiotic used to treat the bacterial infection, and it is for bacteria. So in 1945, yeah, the atomic bomb was dropped on Hiroshima, a city of Japan, by the America. Then the 1949 Anders, Wellers, Robbins grow poliovirus in the tissue cultures. Guys, this is a big thing, okay? This is the poliovirus and this is the human tissue culture. You guys, I don't think would see the importance of it, since human tissue culture means that you are culturing human cells, right, or human tissues, and after culturing that, you are even infecting it with the virus, and that is poliovirus. People should know that the poliovirus does cause the polio, and the polio is a neuro disease, right? Like, it affects the nervous system, and the physical appearance is quite the morphology changes in this disease. So in 1950, Korean Wars began. I don't know if they are talking about the South America or the North America, so let's just search it. The 1950 Korean War. Let's see, what is it about? A conflict on a Korean peninsula from the June. North Korea, supported by this, invaded the South Korea. Okay. So North Korea was actually what do you call it? The starter of this, I think. So they invaded the South Korea and divided country into North and South Korea after the World War II. I think we are coming towards the World War II sooner. And So by the time Korean War was happening, 1953, Watson and Crick proposed the DNA double helix. Okay, so it is by this time that we are just finding out that DNA had the double helix structure. And in 1955, Jacob and Wolven discovered the F factor in the plasmid. Okay, I don't have to search this because this F factor is actually the factor through which conjugation occur, and by conjugation I mean the sexual reproduction of the bacteria. It's not actually the sexual, it's the vertical transfer of the genes, but this F factor is involved in it because it forms the conjugation tube. Let me just show you the diagram so you can understand. Wait, so see here this diagram. So there is a donor and this is a recipient, right? So there are some bacteria that have this F factor and can create the tube, right? And there are some bacteria that don't have that. So it was the first time that this F factor was because of this round thing, the circular DNA, this plasmid. So this was the first time they discovered this, right? So, and after that, after F factor was found, that just shows that there is a way for bacteria to transfer their genes directly from one another and not through generations, right? Normally genes transfer from one generation to another, but in this case it's the same generation and the DNA is transferred from one bacteria to another. It's the vertical transfer, while the genetic transfer, which occurs through horizontal transfer, through generation to generation, is the horizontal transfer, right? So you see the F factor created this pilus, we call that, and through that pilus the tube connects the two bacteria, and as you can see the plasmid duplicates and it transfers to the other bacteria. And now you see the plasmid is transferred and then the tube is formed in this bacteria as well. So this just shows that the plasmid is actually the circular DNA specifically in bacteria is the cause of this F factor, right? That's why it is called as F plasmid, because it has that F factor. So the donor is F plasmid, but the recipient wasn't the F plasmid. But after, not F plasmid, didn't have the F plasmid. But after the gene transfer, so they gained the F factor plasmid. So this was in 1955 that Jacob and Wolfman discovered that. And then what was happening in our world? This is our world as well, but let's just see that in 1957 Sputnik launched by Soviet Union. Sputnik, guys, do you expect me to understand what Sputnik would mean? Sputnik. So let me search Sputnik. Sputnik what? I forgot. What was Sputnik? I don't have the PDF with the Google window as you guys, so I have to search it. Sputnik launched by the Soviet Union. Okay, so Sputnik is a satellite, as it's saying that they launched it. And Soviet Union, let's see what the Soviet Union is. Wait. I don't think we will be getting anywhere if we just look at the images. It was launched in the elliptical low Earth orbit by the Soviet Union on 4th October 1957 as a part of Soviet space program, and it sent radio signals. Okay, so this was the time that we were starting on the early ages of telephone, right? Okay, they allowed to usher in a new political, military, technological, scientific development. When Sputnik launch was a single event, it marked the start of space age. Okay, so this was, I think, was the Sputnik the first satellite sent into the space? Was the Sputnik first satellite, right? Here it is. We don't have to, why is it not searching? Yes. Oh, guys, did you learn something new? Sputnik 1 was the first satellite, artificial satellite. So you guys know what natural satellites are, right? These are the satellites that revolve around the Earth, and they are not man-made like Moon. So if someone didn't know that. So, okay. we are here, finding out the effect of plasmid, and there they are going into the space. Guys, the pacing is not normal. Pacing is not normal. We are still stuck at the microbiology labs, and they are going into the space. They already started to go to the space after launching bombs and wars. Guys, there is wars everywhere. It's like our world is conflicted with wars and physics, or the lights technology, etc. Okay, and here we are studying this. Anyway, in 1959 the yellow develops the radio immunoassay. So let me tell you what the immunoassay means. Okay, guys, by this method we actually use the antigens, or sorry, not antigens, we use the antibodies to find out if there is any antigen in the present sample, in the the sample we have. So how do we detect the antibodies, like, right? So how would we detect it? It's by attaching some external factor to it, and I think in this case it is the radio one. I think I'm getting wrong somewhere. Wait. I think I'm mixing it with something. Let's search it. I think I am getting it wrong. Wait. Highly sensitive technique that measures the concentration of substance in a sample by labeling them with a radioactive isotope. Okay, we are not wrong. Right. So you are concentration. Okay. No, wait. But it said the immunoassay, so of course antibodies are going to come in. I think they are. I wasn't wrong. Yeah, guys, look at this. Oh, yeah. We will see what happens from this diagram. So here we go. Like, look at this. These are the antibodies, these Y-shaped things, right? And these are the antigens, or the substances we need to find the quantity of it, right? We put both of these in the buffer solution or in the sample, and these antibodies, they are connected with the radioisotopes, or right? Immunoassay, I think the isotopes are connected with the antibodies and not with the substances. Okay, no, they are saying the antibodies have the radioactive isotopes. So the radioactive isotopes are connected with the substances, and of course they will be detected by the same process like the luminescence, right? You put something luminescence that produce light, and then you detect it by the photodiode or things like that, optical detectors that would detect that light. In the same case, they will be detected by some radio detectable material, and after that you put the blood or something and antigen, right? And they are labeled. And why do I feel like I said something wrong? Wait, okay, let me see. Here, this in this diagram, this is radio labeled antigen, and this is just antigen, and these are antibodies. Is this to detect they said that this is to detect the amount of the substance, right? The amount of the substance. So the substance should be this antigen or this antigen. Bound fraction. Yeah, I think they both connect to one another. Antigen, antibody, radio label antigen. Antibody, antigen. Antibody, antigen. Free fraction, bound fraction. Bound fraction. Yeah, I think these two connects with one another. Some antigens bind with the antibodies, right? Some antibody, and some of these not labeled antigens bind with another. And after that, the antibodies that are locked, they actually bind with one another, and so they get separated, right? And these are the free sections. Yeah, I don't know if you got it, but these are the radioactive antigens, the primary antibodies, and the unlabeled antigens. See, now this is okay. Now this diagram is okay. The above ones weren't doing it. So these are the primary antibodies, and they have the areas for the attachment of the antigens. Now there are some antigens that are radioactively labeled, and there are some that are not, and both of them bind to the antibodies. See, this side of it is attached with the non-labeled, and one side is attached with the labeled ones, and they are detected by some receptors. But I think we are not getting it with that diagram, so we should, at the last time, should go to the often used to how it works here. A known amount of protein is labeled with a radioactive isotope. The labeled protein is mixed with the antibodies specific to that protein. The sample is added, and any antigen in the sample competes with the labeled antigen for binding to the antibody. The bound and unbound antigens are separated. The amount of radioactivity in the unbound fraction is measured. The level of the radioactivity is proportional to the amount of antigen in the sample. I think I'm getting it wrong somewhere. The level of radioactivity is proportional. So the amount of radioactivity is unbound fraction. Okay. So the bound materials are not measured. It's the amount of radioactivity in the non-bounded fraction, so unbounded fraction. Okay, so again going towards the diagram, it's the free fractions that we measure, and by free fraction we search for the quantity. Again, it becomes the same amount, like we would assume that half of these are attached to the antibodies and half of these are attached to the antibodies. So in free fraction we should just double them, right? Like if we are detecting 2 radioactive labels, so we should just assume that other 2 that that this should be doubled, because somewhere they are going to be attached to the antibodies. I don't know if I'm making sense. This is stupid. I think I am not getting the point. Sorry guys, I'm taking long, but I won't let it go until the labeled protein is mixed with the antibody specific to that protein. Antigen in the sample competes. Guys, so I'm not getting the point of them attaching to the antibodies. Like I get it that they are going to measure the unbound, right? Okay, I see. Why do they need to compete, right? Like I'm not getting the point where they are going to compete. The competition occurs because both a radio-labeled antigen and the unlabeled are added to the solution contains specific antibodies, and they essentially fight to bind the limited number of antibodies. The more unlabeled antigen present in the sample, the less radio-labeled antigen will be able to bind, allowing for the quantification of the unlabeled agent's concentration by the measuring of radioactivity bound to the measuring the level of radioactivity bound to the antibodies. Guys, I still don't get it. They just said that. Okay guys, I'll tell it to you straight. I think they are messing the point somewhere, especially this AI. Somewhere they say that we are going to measure the free fraction, right? That is not bound to the antibody, and here they are saying measuring the level of radioactivity bound to the antibody. Okay, so this is nonsense, right? This is totally nonsense. We understand what competition means. Like if we add the radioactive label ones, they are going to, radio label, what do you call it? Okay, now I get it. Okay guys, this is the last time I'll be explaining this because I get it finally. Okay, so we have antibodies. We have antigens that are labeled, and we have antigens that are not labeled. So we don't know the amount of these antigens, right, which are not labeled, but we know the amount of these antigens which are radio labeled. So we also know the amount of antibodies, like the total amount of antibodies is equal to the amount of radio labeled agents, right? So in theory, if we exclude these antigens which are not unbound, These radio-labeled agents should completely fit with the antibodies, and we should not have free fraction. We should have completely bound fraction, right? This is what should be done. But when we have antigens and they are structurally similar, right? But the antigen is dominant, not labeled one, unbound one is dominant. So when they compete, the unbound ones bind and the radio-labeled one leaves, right? So the more these unbound antigens are present, the more or less these radio-labeled antigens are going to bind to it, right? After that, we know the amount of radio-labeled antigens, right? So we will know that how many antigens in the bound, the bound antigens are in the bound fractions, and the how many bound antigens are in the free section. So with that, when we calculate how many went to this side and how many went to that side, we can find out how many antibodies were captured by these free antigens, and we can finally quantify it, right? So guys, do you get it? We are actually going to measure these, okay? The antibodies that are bound with the antigens. And as you can see, bound fraction would have some of the radio-labeled antigens, and we can find out, if the quantity is high, there is no way there are any radio-labeled antigens leaving here, because they are super competitive and they can replace these. As you can see, this guy is replaced with this one, with this bound antigen, not bound antigen. So yeah, we measure this and we minus it to the total amount, and we will know how many are attached to the antibodies that we know. So this was Yellow develops the radioimmunoassay. He developed such a complex method. Then we are at the 1961 first human in space. What is this? We launched the first satellite, not that. Yeah. What? Shut up, you can go. It's not what you think. In 1961, first human in space. Sorry for my foul language. There was some, donkey here. So in 1961, first human in space. 1961, Jacob and Monod proposed the lag operon. So when did we send the first Sputnik into the space? It was 1957, and soon after the human went into the space in 1961. In 1961, okay, so they proposed the lag operon. Guys, lag operon is the cluster of genes that work towards one goal, and that is the digestion of the organic compounds to produce lactic acid. So this is the basic operons. Bacteria has these operons where there are the cluster of genes that work towards one goal. And for example, there is also tryptophan gene. Let me just show you what genes are involved in the lag operon. See, these are the regulatory type, and these are the Y alpha, ZY and A. So these are the genes, and these are the regulatory genes. So when there is the lactic acid present in the compound, the bacteria stops the production of lactic acid. But when there isn't, the operon is active and the messenger RNA is being transcribed, and then they are translated into the proteins like production. And these are the enzymes, like Z is for the beta galactosidase, and this is for permease. And what? Permease? No, this wasn't this. Permease, yeah, it's permease, not fermiase. Is it me that find it F? Yeah, this is P. And then this is the transacetylase, right? These are the repressor, and this repressor is actually the lactic acid. Anyway, let's go forward. And this is in 1962 first quinolone synthesized. We already know that quinolone is the antibiotic. And what antibiotic is it? It is the DNA inhibitor antibiotic. And this was synthesized already in 1962. Synthesized. Do you get it? Synthesized. It wasn't natural. And how it works? It works by inhibiting the working of DNA polymerase enzyme in one way or another. Now, the 1962 Cuban Missile Crisis. Cuban. Was the missile launched at someone? Cuban Missile Crisis. What happened? What are these? Wait. Again, the Soviet military balled up in a 13-day confrontation between the United States and the Soviet Union in the 19. It was pivotal moment in the Cold War. Okay, but it is crisis, right? 13-day confrontation between. Okay, so it wasn't a buddy. But for what purpose? The crisis lasted 16 to 28. Confrontation is widely came to escalating into full-scale nuclear war. This is no joke between United States and Soviet Union. So, when two superpowers came closest to the nuclear conflict. By the way, what are these images about? So this is Cuba, USA. So one side is the Soviet. Cuban. What does Cuban stands for? Cuban stands for, sorry for the lack of my knowledge, but it's that, you know. Oh, sorry. I just searched it. I just read it, that these are the two, Soviet Union and these. Okay. No, I didn't read it. What is Cuban? What is Cuban? In Cuban, I must be dumb. I think people would know that. Cuban refers to the island nation where the Soviet Union secretly installed the nuclear missiles. I'll just say missiles, bringing them dangerously close to the United States and causing major international standoff during the Cold War. I think these events are just leading to the World War II, and if this is going to go happen. So here the natural war was coming, the nuclear war, and there we are just, I mean this is dangerous as well, like this is disturbing the DNA, the basic of life. And then in 1969, Neil Armstrong walks on the moon. Right, this is Cap. Did we really walk on the moon? But if they are saying, that must be true, and who knows. In 1970, Arber and Smith discovered the restriction in endonucleases. Okay guys, do you know what restriction endonucleases mean? These are the enzymes that cuts the DNA, and nuclease, you would understand nuclease stands for the enzyme that cuts the nucleic acid. Okay, nucleic acid, and in this case, of course, this refers to the DNA. Since I think they are double stranded. And then endo, there are some enzymes that cut the DNA at the the outside parts, and there are some that cuts in the inner sides. The one that cuts the inner parts of the DNA, they are endonucleases, and this cutting part refers to the restriction. So they discovered the restriction endonuclease. Let's see what kind of restriction endonuclease did they discover. 1970 restriction enzyme nuclease discovery. What was it? Let's see, what kind of enzyme did they? It is characterized the first type 2 restriction enzyme, HIND2. Okay, HIND2. So I didn't know that HIND2 was the first restriction, but they are saying this is the type 2 restriction enzyme. But then there was the type 1. Randomly away, yeah. Wait, is the HIND2 the first? Is HIND2, guys, this is the scientific name of the enzyme, HIND2. Haemophilus influenza, IN for influenza, and this D is for class. So is HIND2 the first endonuclease enzyme discovered? Yes, HIND2 is the first discovered restriction endonucleases, and after discovering a lot of restriction endonucleases, we found out that there can be type 1 restriction enzymes and there can be type 2 restriction endonucleases. Sorry, you can say enzyme. It's not that such a big crime to say it enzyme instead of endonucleases. So this is the first endonuclease, and they are saying that this lays the work for the future DNA technologies, recombinant DNA technologies, because now we can cut the DNA. So if we cut the DNA of one species and we cut the DNA of another species, we can combine them, and this lead us to the DNA recombinant technology. And then what happens? Let me just enlarge it a little bit so we can see it. Within 1970 this was happening, and Vietnam War was ending. Okay, how many wars are we going to go through? Vietnam War, Korea, America, Germany. Now this lasted for which and which, between North Vietnam and South Vietnam, with the United States supporting. Again the United States. What the heck is happening with this United States? The war was a part of Cold War between United States and the Soviet Union. Okay, they are doing the Cold War, and now they involve the Vietnam in that, and of course the United States sided with the Vietnam. And who won? Or let's see who won and who lost. And why did the US lose the what? They were supporting. Wait, I'm not getting the point here. Who won this? Marked the end of the now North Vietnamese People's Army launched the Spring Offensive in March. The South Vietnamese Army of the People was cut. So the South was defeated and the North won, just as in Korean War, I think. One won and one lost. Then the 1977 WUS divides the prokaryotes into bacteria and archaea. Now after discovering a lot of bacteria, as we have seen in this landmarks, there comes the WUS that divides them. Are you guys saying that all of these are bacteria? No, we are not considering them bacteria. We are saying that some bacteria are different. So how about we name them archaea? And I think I don't know on what basis WUS divided them into bacteria and archaea. Like, did he study the ribosomal RNA of bacteria and archaea? Like, the only thing that is drastically different between bacteria and the archaea is the ribosomal RNA. So on what basis did the WUS? On what basis? Wait. Why is it going down? On what basis did the WUS divide divided the bacteria based on the significant difference in their ribosomal RNA? So of course. So it is because of the difference in the ribosomal RNA. I think we are in the future now that where we can study the, on the molecular levels, that is the bacteria and, sorry, not DNA and RNA, yeah. So yeah, of course it is 1977. What am I thinking? We are already near to the 2000s. And this history ends on the 2005. 1971, 3 Mile Island disaster. 3 miles? How much is 3 mile? 3 mile. Three Mile Island nuclear incident. So this is the nuclear incident as well, just as the previous happening between the United States and the Soviet. A partial meltdown of the Unit 2 reactor at the 3 Mile Island. I think before that people weren't that concerned, or people weren't that aware of the disaster that a nuclear weapon can cause. And this is 1979. This is very recent that it happened. And so what happened next? 1979, insulin synthesized using recombinant DNA. Okay, so they just found out the endonuclease, and 1970, and they finally started working on it. So in 1979 they found the recombinant, they produced the insulin by combining the DNA. And what happened next? Smallpox officially declared eradicated. I think the vaccine finally started working. Okay, how does the recombinant DNA work? Do I have to explain? Just as I said, recombinant. The DNA in the bacteria was combined with the DNA from the human cells that has the genes for the insulin. And after combining our genes with the bacterial genes, I think this was E. coli, the bacteria, which produced the insulin. And then the patients with which has hormonal problem with insulin, what do you call that, diabetes, yeah, is it sugar problem, yeah. So they were injected with the insulin, and then I think they are temporarily safe to have the glucose levels and eat something they want. In 1980, development of the scanning tunneling microscope. Okay, so we just discovered and developed the electron microscope, and now we are making the types of the electron microscope. Like this one is the STM, scanning. Okay, there are two types. One is the scanning, and like the one that sees what do you call it, wait, instead of me blabbering about it, let's just read it. SEM. Okay, here they are scanning electron microscope. So they are high resolution images, right? And I think they don't see the inner side. Okay, no. How was scanning microscope and TEM? Wait, transmission electron microscope. How is it different? I forgot which one sees the surface and which one sees the inner side. Detecting, we are not talking about, okay, passes through. Okay, so this one TEM is for the internal structures visualization, and SEM is for the outer structure visualization. But this one is not scanning electron microscope, this is scanning tunneling scanning tunneling microscope, right? How is it different from the normal electron microscope? High resolution images, SEM, images of the surface at the atomic level. No. We are not talking about this. Versus SEM, how are they different? One, okay, tunneling microscope offers significantly higher resolution than the scanning electron microscope, allowing it to. Okay, but why is it more high level than the SEM? Like you see this, there are electrons in the SEM. Okay, here, down, see, wait, let me fix my mic. Here, button. STM using the quantum tunneling current between a sharp tip and a surface, while SEM uses focus a beam of electron. Okay, so this is using the electrons while this is using the energy, right? So of course it has something going on back on it. So why do you need this? Okay, okay. Turn this off. I was just checking if it was working. So in 1980, we developed the scanning tunneling microscope. But where was the scanning? No, sorry, electron microscope. Okay, it was far in back. I thought it was near. No, 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 no. It was, yeah, in 1933. Okay, it's not close. That's 1933 and this is 1980. And what? First home computers. Okay, of course we are going to have computers since we sent people to the space and the moon and the satellites. So why can't we have computers? Okay, where am I going? First home computers. Home computers. So they are not talking about the history of the computers from the start. They are just directly jumping to the home computer. I think they were big enough. Like, the CPUs. You guys, do you remember the CPUs with big backs? First home computers. Wait. Let me see. Here. What? Is this how it looked? The screen is smaller and it is blue. Okay, what the heck was I expecting? But it is colorful. Look at the quality of this. At least the keyboard and the mouse is the same. Okay. Wow, they look alien. I thought after, enhancing we will come to that, but no, after enhancing it we are looking less alien than this one. Sorry for my extra talking. I just can't study without talking about something else. Oh, finally, we are at the last page of these landmarks. Let's see if something big happened after the 1982, since it's the era that is closest to us. In 1982, recombinant hepatitis B vaccine was developed. B vaccine. So it was, but what is recombinant vaccine, right? We know the typical vaccine. For someone that don't know what vaccines are, they are either killed viruses or the parts of the viruses, or just a little bit heated, not completely killed, weak viruses. They are injected into body and our body responds and develop immunity against it. But what is recombinant, right? Like, this is the first time. Recombinant vaccine. This is the first time I'm hearing about the recombinant. Is it just me? Here. Isolation of the S gene from HBV, insertion into the yeast, multiplication of yeast cell, extraction and purification of HBsAg, absorption on. Okay, so just as I said that we inject the gene, or the genetic particle into the cells for us to develop immunity. So in this case, I think they couldn't put the gene directly into us, like inject it. Maybe it would have caused problem. So they first put it in the yeast and combined it with the yeast DNA. So after multiplication, more and more of yeast cells were produced, and then the DNA of these yeast cells were extracted and purified. And yeah, this was combined with the adsorption. Sorry, I said absorption first, but this is adsorption. So yeah, they were attached on it. Yeah, they need something for us, for it to go inside our cells, or for it to induce immunity in us. So okay, so this was recombinant vaccine. I hope you guys understand it as well. And what happens next? Before this vaccine was developed, first space shuttle was launched. Guys, are we not done with this space? First, looking at this, where are we at space now? I think there was some kind of telescope sent into the space for it to get the images of the further and further galaxies. Okay, sorry for me chatting. What was I searching? first shuttle, okay, launch to go into the space. We need to look at it. What? Guys, this was not what I was expecting. Don't tell me this is the first space shuttle. Don't tell me it was this big and advanced. I thought it would be something smaller. Right, it's a complete aeroplane, but what was its purpose? Wait. Name the space shuttle Columbia. So we are not talking about this. Of two Commander John. Okay, so it has people in it. It had people in it. Sorry, not has. Okay. So this was the Sputnik, and this is the first shuttle. And the first person, pilot Bob Crippen and Commander John Young. Here, John W., but this is Young. Okay, we are not going to go deep in this. You guys will be saying that, are we studying microbiology or are we studying the history of the world? Anyway, here is the, where were we? In 1983 to 1984, the isolation and identification by the Gallo and Montagner Mullis developed the PCR technique. Okay, I think this is the era of DNA kind of things. Like this was involved in the DNA, and previously we studied about the restriction endonucleases, and we talked about the what? I forgot. What else did we talk about? Yeah, here we synthesized insulin using the recombinant DNA. So this era is about the DNAs. Like even in the microscopes we are going, okay, so not the DNA, this is the era of molecular microbiology. We are going deep into the, why change the page number, guy. Okay, here. So the PCR, polymerase chain reaction. So should I explain it? I think people listening to this. So guys, PCR is the polymerase chain reaction, and polymerase is an enzyme that catalyze the, or do the synthesis of DNA, and this is used for the multiplication of the DNA we have. So this is the first time, sometimes we found out a bacteria and that is so low in number, but we need its DNA, and if we just centrifuge them and isolate their DNA, so that's going to cause us a problem. What if we destroys this DNA? How are we going to save the DNA? Like it would be finished if it's a rare DNA we found out and we don't have any way to preserve it, like multiply or quantify it. But then they developed the PCR technique, where we can amplify the amount of DNA into millions and billions of copies. Okay, so there is a process about the PCR, but I think it is not the time to go into that because we are just studying the history. I'm more interested in looking the PCR machine. What did the PCR machine, the first one, I am more interested in looking at that than finding out what the process was or what. Oh, so is this how it looked? What are you talking about? This is exactly what it looks like now these days. Like look at this. This is PCR. We put this, and the rest of the process happens on its own. We put the polymerase. Okay, but this would mean that we already have the polymerase enzyme, and that was the tag, the first one that we isolated from the bacteria that lived in the extreme condition, aureus or something. Some name was something like that. So they are not showing us, I think, the, what do you call it, the ancient pictures. I wanted something, the first one, PCR. Okay, I think it looked something like this. But guys, this is just in 1985. How do we develop something like this? But then again, we had the home computers. Okay, so the technology wasn't that bad at this time. But then again, it's so late. Why do we have technology like this? I thought things, but then again, whatever. why am I obsessing with the technology of that era? Okay, so they developed the PCR technique, and by that they would mean that they had the polymerase enzyme and they had the machinery, and what DNA did they, amplified by the PCR technique? It was HIV. HIV is a small virus, and we need multiple copies of DNA to study what the pattern or sequence are. This is identified. It's what is the relation between PCR and identification, and it's that which we can identify microorganisms by studying its DNA. And for us to study that, we need the DNA to be in high amount, so we can study more and more if we need it. And of course, for sequencing, we need layers on layers of that, and we need to cut it with the endonucleases, so smaller, and from smaller to larger portions, pieces of that DNA comes and they align themselves, and then we add the radioactive substances with it, and then we detect the radioactivity of them, or luminescence of them, in case if we put something fluorescent with that. And so yeah, in the end we detect it, we identify the DNA sequence, and then we search that what is this sequence about. Like, I think in 1983 there wasn't a library that large which had all the DNAs in it, right, the sequences. But like our own human DNA was, I think it was 2002 that it was done being sequenced. It is, I think, the largest DNA maybe in the world. No, I don't think it's the largest. There, like mango has, we have 40, what do we call it, 23, 6, like we have 46 chromosomes, and the mango has 48, I think. Sorry if I'm wrong, don't come after me, but I think there are other things that has more genes than us. But I'm just talking about the sequencing talk. Like, we can sequence the DNA, and then we can identify what this bacteria is about, what DNA, whose DNA is this, or who does it belongs to. Like, for example, we found some random hair on the path, and we need to find out whose hair is this, and who it belongs to. We can just do the PCR, and these days it's easy because we can just put the sequence in the computer, and whoever it matches with, I'm talking about species. So we can find out if it's human hair or monkey hair or rat's hair, right? My pronunciation may be not right, so it's the spikes that grow on a hair. Not hair, head. So then it's the 1982 first artificial heart implanted. What the heck? Okay, I know we are technologically advancing in the microbiology, but what? First artificial heart implanted? Okay, we are going to Google for this. How did the first artificial heart looked like, right? Oh my god. But then I think they cloned it, since we already have seen the human cell tissue culture, so it's no surprise that they can clone a complete organ. Not they advanced from tissue culturing to the organ culturing. And is this how it looked? But I thought it was a biological. This is really artificial, made from plastic and some metals, etc. What the heck? But did it work fine? Okay, I think skipping some steps, and I don't think as soon as they created the artificial heart they implanted in someone. Okay, so I think at this time they just finally studied the structure of the heart to its core, and they made the artificial heart. Yeah, of course. How would it work, the artificial heart in our bodies? What? Okay, so I was going to the culturing side. Of course that happens as well, and that will be happening in the future. Like, it's just not succeeding, and it may be just complex, but that happens, right? It happens. Okay, but they were really talking about the artificial. Okay, I mean, we don't call the cultural one natural as well, so that's why my mind went to that side. So this was in 1982. Okay, okay, we are going to space. We are recombining DNAs, and we are amplifying the DNAs, and we are producing the artificial heart. Oh no, they are saying the implantation of the artificial heart. So it was working and it was implanted. So I think they just didn't reach the area where they could take the heart from someone else and put it in the humans. So that's why they had to make the heart. These days we can take the heart from someone else and just implant them, or we can just make the clone of the heart, but I think the succession rate for that clonal heart is low. That's why they focus on implantation from the human to human. So now we are going to 1985. I was jumping to 86, but then I see 85. Here it is 85. The Gorbachev becomes the Communist Party General Secretary. It's a Russian general, I think. Mikhail Gorbachev. I was played in my childhood that this was a ghost and they will hunt me in the night, but it looks like it was just a person, and they just liked the name and they wanted to scare me off by the name. So it was a Russian, yeah, it was a Russian politician and a statesman who served as the last leader of the Soviet Union. Okay, so we are still at the Union part. Okay, I understand it was a great person, and I think after that Soviet Union would have been collapsed. Yeah, it's written with that. In 1985, Gorbachev, and it was the last general. And then in 1991, after a few years, Soviet Union collapsed. First vaccine developed by the genetic engineering approved for the human use. So for until now we were developing vaccines, using the same materials that are natural. But this is the first time by genetic engineering vaccine. Like, we didn't know the succession rate of the genes that would, we with our hand cut it and then combine glued, and no one knows that those genes would succeed in the human body, right? I think they practiced it on the, guinea pigs or maybe rats, or for them to know that, yeah, it would succeed. So it was in 1986. Like, I think the technology has gone far better in the late 1900s. So 1990, first human gene therapy testing begins. So they are already jumping to gene therapy. I don't think even in 2025 we are doing the gene therapy. What are they thinking about? Testing? Human gene therapy testing? 1990, and this is 2025, and we still don't have the human gene therapy. What the heck are they talking about? But then again, it's the molecular, just as I said, it's the molecular microbiology era. So we are, of course, testing human gene therapy. I don't know if they succeeded. Of course, they are just testing. Okay, let me a little smaller. I mean, yeah, whatever. 1992, first human trials of antisense therapy. Okay, are they thinking that humans are test subjects? Are they not focusing on animals, like they are practicing the human gene therapy on it? And then what was this one? This one was the antisense therapy, and this is also related to nucleic acids. I can sense it. Like, it's the, I think, messenger RNA antisense therapy. There are messenger RNA and antisense oligonucleotides to treat a variety of diseases, including cancers, genetic disorder, and neurodegenerative conditions. Are short synthetic DNA-like molecule. Yeah, just as I said, antisense oligonucleotides. You can see these are nucleotides, but I think there is a carbohydrate attached to it. That's why it's a synthetic DNA molecule. It's a synthetic because No DNA with anything like polypeptides or carbohydrates can go into our cells and work like normal things. That's why they had to synthesize it to change it to something that can go into our cell, and that's why they can bind it to messenger RNA sequence. Normal DNAs don't bind to the messenger RNA sequences, okay, blocking the cell's ability to use RNA to make proteins. I think this is especially for the cells that has been infected. That's why they are saying that it is used for cancers and genetic disorders. Like, they don't need that gene that are causing disorders. And the same is neurodegenerative conditions. And antisense, there is a sequence. We always started from 5 to 3, 5 prime to 3 prime. I think people would know what I'm talking about. There are 2 chains of DNA, or not chains, strands, and one is sense and one is antisense. So I think the opposite one is the antisense, right? The one that is complementary. So yeah, they use that kind of, and because it's binding to the messenger RNA, that's why they use the DNA-like molecule, right? Not directly the DNA. And antisense means it has 1 strand only, not 2 strands, even if it is a DNA. We guys have single-stranded DNA. We don't have just double-stranded DNA. It's not the double strands that make it different from the messenger RNA. It's not the strandings that separate the DNA and RNA. It's just the nucleotide, because messenger RNA has uracil in it and DNA has thymine in it instead of uracil. So what happens next? We have, okay, and before antisense therapy, the Soviet Union collapses. And after that, bang, 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 we are still at genomes. How about we leave these genomes alone? 1995, chickenpox vaccine approved for US use. So what was the vaccine before that? I have memory of what? What? Did we went somewhere wrong? Okay. Oh yeah, it was a hepatitis B virus. Yeah. By the way, what was the 1986 vaccine? Right? This one. First vaccine. Wait. We have to search this. What was the first genetically engineered vaccine? What was the first genetically engineered? Whatever. Forget my spelling. I am trying to write it fast just to not bore you guys. What the heck? It was the same Recombivac's HB vaccine. Okay, so first they develop it and then they put it in the human. Okay, so this is 1986 and that was 1982. Okay, it's the same thing, man. They had to make it like it's something else. Okay. So, okay, sorry. Look, it's not your fault. It's my fault. It's my brain's fault for not understanding it. Haemophilus influenza genome sequenced. Sequencing. See, we have PCR now, and now there will be sequencing, and that's all. So for now, Haemophilus influenza genome is sequenced. In 1996, the Methanococcus janischii and yeast genomes are sequenced. But don't worry, Methanococcus is not the only one, since just as I told you, in 2000s we will be having our own genome sequenced. In 1997, what happened? The largest known bacteria, guys, remember it, it's the largest known bacteria these days, okay? Thiomargarita namibensis. Wait, is it still the largest? Let me Google it. What is the largest bacteria in the world? Images. Okay, so they are saying it's the Thiomargarita namibensis. It's a bacillus. Yeah. Okay, it's a chained bacillus. So it's the same. We have Thiomargarita namibensis. Even though this is the 7th edition of the book, We still haven't found a bacteria larger than this. And this was in 1997. I think when they discovered the bacteria, at that time I didn't think it was the largest. No, wait. If it is still the largest bacteria, of course it's going to be the largest in 1997 as well. Yeah. And in 1998, water found on the moon. Guys, this is exaggeration. Okay, of course there are going to be drops of that, but don't tell me we went to the moon. No, 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 no. I'm not believing it. There is just no way we went to the moon. Who believes it? Raise your hands. 2000 discoveries that Vibrio cholera has 2 chromosomes. Okay, I think till this day, this is what? This is bacteria, and bacteria usually have 1 chromosome. Understand that? But in 2000s, we found out that these have 2 chromosomes. So this is nothing big. I mean big to check in the Google, right? We understand it. 2001, anthrax bioterrorism attack in the... What? Okay, I for a second thought, what is the world history doing in our microbiology history? Okay, it says anthrax, okay, it's a bacteria. And this anthrax, we had we had it with you. Anthrax, please leave us alone and please remain in the history, okay? We already studied you. You don't have to be used for terrorism. In the New York. Okay, so the coronavirus isn't the first bioterrorism. Guys, is it a terrorism? Is coronavirus really, implanted, technically, and it wasn't natural? Okay, so it wasn't the first time. Humans are strange. So they used the anthrax attack in the New York, Washington D.C. and Florida. Yeah, it's America. Yeah. Wait. What if I'm wrong? Is New York really in the America? Don't call me dumb. I don't know. Is New York? Guys, you just don't know that my America continent. You guys just don't know that I don't know a thing about the world. What is this? Where's New York? Here. This is USA. This is Canada. So it's North America. Okay, understand. So 2001 answer. Okay, we are at the end finally, and again this America is involved. Guys, the people that lives in America, they should read this history book, right? They should read it. There's America over and over again, and then here is World Trade Center attack. Okay, we already had one time that the stock market collapsed, and now we have Trade Centers attack. It was 2001. Second war with Iraq. What was the first Iraq? Okay, I'm jumping. I shouldn't. So World Trade Center attack, and I have no. We need to search it. World Trade Center attack. World Trade Center attack. What is this? What? Don't tell me this is 9/11. Huh? 9/11 attack was World Trade Center attack. I thought it was, wait. How many trade in World Trade Centers? 2000. Well, it's less than the corona, right? Yeah, why do they make a big deal of it? Oh, sorry for the humanity, for the humans that died. Sorry for you guys. But then again, it's not that as big as corona. But still, wait, what is 9/11? Okay, guys, even though I have heard big deal of 9/11, I am still me. I still haven't learned what this is. The September 11 attacks, also known as 9/11, were a series of coordinated terrorist attacks on the United States on September 11. Guys, it's about time. The United States is going to be attacked. Didn't we already study a lot that US has been involved in a lot of wars? It's normal for them to be attacked. I mean, it's not a good thing. No violence is a good thing. I'm just saying that US should have seen it coming. The attack were carried out by Al-Qaeda, Islamist extremist group. So maybe, but what was the purpose of them attacking? The US. I mean, the whole world is there. Why the US? Wait, why did Al-Qaeda attack US 9/11? Okay, forget it. I am always writing wrong. Motives for the war complex and included religious and political factors. The whole world is, causing problems. It's not just the US. Some say the attack was a religious act to restore the moral order to the universe, but why US? Was US immoral? Al-Qaeda messages to Islamic audience were theological and did not guys, it's a dark history. Even the Wikipedia is no, it's AI overview. Why am I reading the AI thing? Were carried out by 19 hijackers of the Islamic militant organization. Series of okay. So guys, if you know in detail what actually is the 9/11, you have to enlighten me about this, because I'm not going to search it even more than I have already searched it, because it's talking about may and could and can and might and may, and these aren't certain words, and I'm not going to deep dive. Okay, dive deep, sorry. And okay, so infected polio were synthesized from the basic chemicals. So in 2001, 9/11 was happening. Why didn't they write it 9/11, and why did they say World Trade? Were they trying to be simple? Moral? Infectious polioviruses synthesized from the basic chemicals. Why are we synthesizing poliovirus? Why are we synthesizing it? And it's even infectious. It's not even the healthy ones, that they don't cause diseases. Why didn't we write the human genome sequencing in our 2002, which I was saying before that? Why do we have to think about the poliovirus? I think the one that wrote the book thought that, yeah, we are done with the genes. The people studying about genes, they will be thinking that, is gene all about microbiology? So why not put the poliovirus? Okay, from basic chemicals. Okay, I don't know how they, produce the poliovirus from basic chemicals. Should we study how the infectious poliovirus is? Okay, how the infectious polio viruses were synthesized. Okay, let's see what the Google have to say about this. Assembling the nuclear pair and the DNA sequencing, then transcribing into mRNA, and finally, okay, so it's not that big deal. So they just took the, okay, so they knew the sequences of the polioviruses, just as we know that they studied the sequence. So they put the sequence into the PCR, and they put the nucleotides and the enzyme, etc., all into the PCR machine, and then the PCR machine just created the DNA and then amplified it, and then of course we put the RNA polymerase that converted the DNA into RNA, and then we put ribosomes, and they translated RNA into a polypeptide. Okay, these polypeptides, of course, the structural proteins or maybe some kind of enzyme or something that would be needed for the virus, right? Okay, yeah, so I get it. But where are chemicals in this? Do they call the nucleic acid base pairs or things the chemicals? Of course there are enzymes. Is enzyme considered chemical? I know there are pH buffers involved in this and some glue involved in it, and yet temperatures involved, but they call it chemical. What? They're messing around with me. I don't get the basic chemical part. 2003, second war with Iraq. What was the first one? Why they skip from first one if they are going to mention the second one? Wait, sorry. This is going black. What? Guys, I opened the wrong window. Give me a second. How do we close it? Yeah. Okay. So the second war. What is the first? What is the what? Wait. What is the first and second war with Iraq? Now Iraq did what? What did Iraq do to deserve war, and who was it going against? The first war in Iraq was 2003 invasion of the Iraq, and the second was the Iraq War, which lasted from the Iraq War was a part of global war on the terror. Okay, Iraq is a Muslim country, right? And the 9/11 was the, the Muslim or Islamist, they said. So is it because of that? What the heck, US? What the heck? You here again? What are you doing? Invading the Iraq, going to destroy the Iraqi weapons of the mass destruction and the dictatorial rule of the Saddam Hussein. Okay, I get it. This second invasion, I get it. But what was the first one with? After US invasion in the 2019 is often known as the first Iraq War. Okay, so it wasn't first, but after the second, I think the first one was also with the US. Because why would they call it the second if it is just a war? 2000 SARS outbreak in the China. Okay, guys, does this name sound familiar? We know the COVID-19. Its full name is SARS-CoV-19, right? It's SARS. It's the same class. So are you telling me that in China this outbreak happened before, and now the people are, should I say it? Will I be banned? But whatever, we are studying science. We should, put everything on the line. So people are saying that the COVID-19 originated from China or something. Are people saying that, or am I just is it just a miscommunication? Anyway, 2003 SARS outbreak happened in China, and then 2005 super resistant HIV strain isolated in New York. We already were having HIV problems, and now there comes the resistant one, which we don't even know. HIV till this day don't have the vaccination or treatment that could completely get rid of HIV. People with HIV develops AIDS, and they are diseased person or immunocompromised person for the rest of their life. If they don't take care of themselves, they can be attacked by other infections very easily. So this is 2005, right? 2005. But the thing is, we are where? We are 2025. So let's go one by one and study the discoveries of each year of microbiology, from Google. So let me just fix the screen first. Let me enlarge it, since what? We are going to, how about this? How do I make it even larger than this? So we are going to search it. Wait, let me put this chapter name in the upper layer so people can just see that what we are studying. And how do we do this? Oh my god, I didn't mean to go to another page. Wait. Is this okay? Okay. Now let me see if it is working. I should just, please give me a minute. I'm fixing this. Okay, is this fine? Yeah. So we are going to go through each year one by one and see what happened in each year in case of microbiology. So top microbiology history in 2006. Okay, so vaccine history is inextricably linked to the histories of microbiology. No, we are not talking about, we are, what is this? Okay, no, wait guys, we're studying the book. Why are we searching on our own? This doesn't go into the criteria of the book reading. Yeah, I'm not studying, neither are you guys studying. We should search something fun, which would be, yeah, let me revise the history of microbiology. History of microbiology for the people. Okay, this is done by AI. Like as you can see, I just put on the Google and then this happens. So let's just went through this, that what were the important scientists. I didn't know the Beecher nickname was going to come here. Okay, so the people that are just starting the history and want a shorter version of the history, let's just summarize the important people. Anthony von Leeuwenhoek, a Dutch draper who is considered the father of the microbiology. Okay, it's just the microbiology, not the modern microbiology. And it's 1670. He was the first to document his observation of the microorganisms. Protozoans. This guy, Anthony von Leeuwenhoek, was the person that started the microbiology. He was the first one to even observe the microorganisms, and it was because there was no one else that designed the microscope. And even if there was someone that somehow designed a microscope, they didn't document it, so we don't know who the nameless person would be that saw the microorganisms even before Anthony Leeuwenhoek. So this guy, so I should show some respect. So this scientist was the first one to document his observation on the microorganisms using the microscope. He designed himself. This is a great feat. He designed the machine and then he observed the microorganisms. Robert Hooke recorded observation of the microorganisms in 1665, including the fruiting bodies of the molds. What? Is there something great? I think there was something. Yeah, Robert Hooke. What they put here, I think this is not interesting one. I will tell you the Robert Hooke. The history of Robert Hooke is with the cells. When he was studying the cork cell, cork means the dead outer layer of the plant. When he was studying those cells, and he studied under the microscope that there were compartments in the bark layer. It means there was a system of compartments, and then he found out somehow that they were separate compartments and they were working alone but collectively in some way. And then he named them animalcules, the microorganisms. So this guy was involved with studying the cork cells of the plant cells. Means he was the first one to study the cell, right? This I think the AI just wanted to say the history of microorganisms, like studying the molds. And then Louis Pasteur, guys. So after this era, a large debate or competition was happening, that about spontaneous generation, as written here, right? Spontaneous generation, that the life arises from nothing, like it arises out of nowhere, like there is no backstory of this. Like if we see food and life would come out of it, like the flies. The people were giving example like meat And you see, if we leave it alone, there is nothing in the meat, and then the flies come out of it, right, after some days. But then this Louis Pasteur did a lot of things, and he was a guy in 1857. Like, he explained the fermentation. People did fermentation before that, like they would make dough, and they would, do things that, but they didn't know that the fermentation were done because of the microorganisms. And the Louis Pasteur told them that it was because of the microorganisms. And then again, I think he was, in the field of food microbiology, so he explained the spoiling and the contamination of the organic substances. And his work disproved the theory of spontaneous generation and established the germ theory. Germ theory of disease means there were eggs, like there were something of life that originated, that, further in, wait. Germ theory of the disease is that there are some smaller particles that we can see, or there, that sprouts the new life. Disease. Wait, guys, wait. I'm blabbering nonsense. Germ theory. That the microscopic organism called germs cause the infection. What? Oh, wait, what? What? The germ theory has then no relation with the spontaneous generation. Yeah, so they are separate things. Sorry, I was mixing them up. So germ theory of the disease. Yeah, we know that microorganisms cause the diseases that can be transferred from one person to another, and this Louis Pasteur said that. And of course, as you can see, his name Pasteur, right? So the pasteurization method for, cleaning the food from the contaminated microorganisms. The contaminated fruit from microorganisms. Louis Pasteur was the first one to develop the pasteurization method, as well as it is named after him. And the spontaneous generation, he did it by, having a flask. Wait, let me show you. Louis Pasteur. Wait, is this it? Why are we going here back? This is not germ theory. So I wasn't wrong. So they are together. He had some meat, and when he heated it, the microbes were killed, and so they didn't cause disease anymore, right? But when he didn't heat it, the microorganisms were present, and so when he applied it on something else, it caused the same disease. But when he removed when he locked the neck, right, when there was no air, of course air has microbes in it, so even if he was heating it, more and more microorganisms were coming and contaminating this. And then what happened? Again, if you even apply heat, and the flask went sideways, so the microorganisms present on this neck area, so of course they are going to be there till then. So of course it's kind of, like said that the microbes cause the disease, and this is combined with the spontaneous generation in a way that, even if you see there are things that nothing is in there, like there is no life, there is no life looking in here, but then again all of a sudden there is a life in it. It's not because there is nothing in it, there are spores in it, right? Even if you heat it and the spores remains, the bacteria will grow. It's the same case with the meat thing, where the flies eggs were there. What am I blabbering around about, guys? What did I do with the chapter name? I think I'm getting tired, so I am getting off track very quickly. Let's go back. We were at the Louis Pasteur. Like, he did a lot of things. Even if the Antony were the father of microbiology, it's not like he did a lot of things. He just found the microorganisms and opened the world eyes to these microorganisms. And this Robert Hooke, of course, he did something like cell. Cell is the basic, structure, functional unit of life. So of course, the name gotten bigger. But I think Louis Pasteur was the first one to do so much in the world of microbiology. Then the Robert Koch established that microbes can cause disease. He deserved the criteria for the establishment. Okay, so now Robert Koch, like he said the germ theory of the disease, right? The Louis pasteurization. But the Robert Koch actually explained us how the microbes can be transferred from one person to another. Like he actually experimented on the disease. Let me explain it by the diagram. So I think it will be fun and you'll understand it more. So Robert Koch, we also call it Koch's postulates, right? And we will see it by diagram here. So there were 4 postulates. I think this is way too blur for you guys. How about this? Is this one okay? This is way too blurred. I don't think you guys will like it. Wait. What I think it's okay, we can see it. So he took a mice that had the disease, right? He studied it under the microscope. He found out that this organism had microorganism in it. He cultured it, and then he took these microorganisms and injected into a healthy mouse, and the mouse developed the same symptoms as that one, which is the death. And when he cultured the blood of this microorganism again, the culture of this mouse again, the microorganisms grew again. So this just showed that if a mouse is ill, if a mouse had disease and it was caused by microorganisms, and if we culture these microorganisms and inject into a healthy person, they will develop the same signs and symptoms as the previous diseased organisms from which we cultured the microorganism. Furthermore, the microorganisms that we cultured from the diseased organisms and the microorganisms that we cultured from, this healthy organism in this unhealthy mouse after, it was injected, it should have the same composition in structure, like morphology of these microorganisms should be the same, because they should be the same microorganism, means there is no change in the microorganisms present in this one and that one. Like nothing went wrong, so that means it can be transferred and it can impact it the same way as it impacted the last organism. So this was the Koch's postulate. Wait, let me just read it just in case we couldn't see it clearly. So now we will lay, right? Presence in the disease of individual. First, there should be microorganisms present in the individuals that are infected, but not in healthy organisms. Wait, let me just make it a little bit smaller. This is actually very difficult. Then the isolation and culture. The microorganisms must be isolated from the diseased individual and grown in a pure culture. Then we should be able to isolate these microorganisms and we should be able to culture it. Then the transmission, we should, like if we introduce these microorganisms, the cultured microorganisms must cause disease, and then we should, we can isolate. Must be, I mean the must is more like should be, not should be like it could be. sorry for going off tracks. Like we can isolate these microorganisms again. It's could, it's more like could than must and should. So the microorganisms are really isolated and it's looking exactly as the original microorganisms that were in the first unhealthy organism. So I hope it made some kind of sense. Where is it? Where were we? So this was Robert Koch's postulate. Then the Ferdinand Julius Kohn. This is the first time I'm hearing your name, guy. Who classified bacteria into 4 groups based on the shapes. Okay. Ferdinand Julius Kohn. Okay. Iwanowski demonstrated. Iwanowski. Disease that caused by the agent that could pass through the bacterial filter. Okay. So he demonstrated that a disease is caused by something that can even pass through the bacterial filter, like it's something even smaller than the bacteria, right? And by that time he didn't know that it was either a living organism or non-living organism, but it was transmittable, and they didn't know that it was virus. So he took the membrane, and like it had the bacteria in it and maybe viruses in it, and when we filtered the sample, the material, when he put it on, organism, the disease appeared again. So like the bacteria was filtered but the disease still appeared. That means there was something else even smaller than the bacteria that could pass through that filter and cause disease. While Bejernik, this scientist actually first cultured the virus, and this was through proper method, and we can say that it was from tobacco mosaic. It was the tobacco mosaic virus, and they cause disease in the tobacco plant, and leaves of the tobacco plant. And after we did the same process as Ivanovsky, we still found out that there was something that after we put the sample on the healthy plant of tobacco, the symptoms appeared again, the diseased symptom. So he said that it was something smaller than the bacteria that could cause the disease. So it was just agent by Ivanovsky, but this Bejernik said that it was the infectious agent. There are a lot of more scientists here, but I think you should remember these first 2 or 3: Antonio van Leeuwenhoek, Lis Pasteur, Robert Koch. And I think this is not enough, so you should know who is the father of modern microbiology. You guys should know that as well. So Antonio van Leeuwenhoek, Robert Koch, and I think this is just not enough. The scientific names which I studied were more than this and were different than this. Yeah, there was a scientist, Alexander Fleming. Where did the Alexander Fleming go? Alexander Fleming. He was the first one to, discover the antibiotic, the penicillin, when he was working with his bacterial culture, and the next day when he saw the bacterial culture was, disappearing, like in an area there was no bacterial culture, he said, what is this? Why is there no bacteria in here? Why is there no colonies forming? So then he found out, there was a fungi, penicillium, and they were producing some kind of substance which was, later named as penicillin, and they were the cause of, stopping the growth of the bacteria. So Alexander Fleming, why weren't you here? And there was one other scientist, sulfa drug. Sorry for me writing, random kind of sentences, but they would understand. Gerhard Domek was a German microbiologist and pathologist who discovered the sulfa drugs. This is a big thing. And specifically what? Prontosil, the first sulfa drug. I didn't know this was the Prontosil. I won't lie. Yeah, I was more familiar with the sulfonamides, right? It could treat the bacteria, but I didn't know that the Prontosil was the first sulfa drug. So the sulfonamide drug is actually more effective, and I think we use it, but the Prontosil is the first one. So then what? Who is remaining? I think this much is enough. This much is enough. And so these were that, and we already studied a lot about the history of microbiology, and we are not going to go back. There are a lot of gene therapies happening, insulin discovered, and double helix structure, plasmid, sulfa drugs. See here, 1935. Wait, what am I doing? You guys can't see it. Here, yeah, the Google is got. Now you guys can clearly see. Nitomek discovered the sulfur drugs. By the way, is the streaming working? Yeah, working. Oh, and I should cross this. Why does it appear every time? See, the Domek discovered the sulfur drug. This is the big thing. Remember who discovered the electron microscope? Prescott, and like Griffith discovered the bacterial transformation. See, Fleming discovered the penicillin. And these are the important ones, which are asked a lot when when you go in front of some examiner and they are taking tests. Like they are just checking the basic knowledge of you. So yeah, we studied a lot. And what was in this thing? Yeah, the first compound microscope. Remember who it was? Jensen, micrographia, and these. Yeah. So this is a spontaneous generation story that was happening. Where are we going? We are not going there. And this is Leeuwenhoek. Okay, so Leeuwenhoek was, the father of the microbiology, and he named them animalcule. Okay, so sorry, it wasn't Robert Hooke. It was the Leeuwenhoek that named the microorganisms animalcule because it resembled the microorganisms he was used to seeing, like more like the helminths and these kind of organisms. Why am I going back? And okay, forget it, we are done with this. And after this we are going to start the discovery of microorganisms, and I think we are going into the detail. Here the names which we just searched are written here, like the important ones, right? So we are going to read this next. So I hope this was some kind of help. I'm going to short these, cut these lives and make a big video, not extremely big, but a video that is smooth and fluid, with no me stopping and pausing, and cover the majority of the landmarks in the history of microbiology. So don't worry if you can't see all the live. I will be making a long video, and it will work fine when you study that. The major points will be covered. Anyway, this was done, and we are still at few pages in this chapter, so it will be a long ride studying this book, but I am sure that we will be great microbiologists after we complete this book. So this is the end.